So what about asthma? This picture shows a gross view of occluded airways. You can see the stars that are present here. Those are the occluded airway. There's an open airway just to the right of the others. You can understand how that mucus blockage would be a big problem. So what happens to the alveolar wall during asthma? So the normal airway has that pseudostratified ciliated columnar epithelium, and it has a little smooth muscle right underneath it. In an asthmatic airway, the entire wall becomes very thick, and that's partially due to hyperplasia of the smooth muscle that underlies the epithelial layer. And also, the epithelial layer can change as well. It can become thicker. It can get other cells that migrate into it. And it also gets an increase in mucus secretion via CNS stimulation. There's inflammatory cells and chronic inflammation due to mast cell secretions in the underlying connective tissue that affect both that connective tissue and the epithelium that overlies it. What does that look like? Well, here's a nice image showing the really large bundles of smooth muscle in an asthmatic bronchiolar wall. Normally this would be a much smaller layer. So this is an example of hyperplasia. And so not only do you get prolonged contraction and closure, or narrowing of these bronchioles, you have so much muscle that they really close off quite a lot, as well as too much mucus too. So here's another image showing on the left pretty much a normal bronchiole, and on the right you can see the very thickened wall of that bronchiole, and that is by both smooth muscle hyperplasia and excessive contraction due to the excessive muscle, as well as some thickening of the epithelial layer too. So the airway is just much smaller in someone with chronic asthma than it is in someone without. Other cells are involved in that process. As I mentioned, mast cells previously. They're responsible for vasodilation and edema of that underlying connective tissue, and that really builds up the chronic inflammatory response. They stimulate eosinophils, and the eosinophils in turn stimulate the smooth muscle hypertrophy. So it's really a chain of events here. The eosinophils are, as I said, responsible for some of that hypertrophy, as well as secreting major basic protein, which damages the epithelial wall. So there's a lot of events going on here in asthma. So, enough about the airways. Let's talk about the actual gas exchange system. What are the components of that? The components of that respiratory portion or gas exchange portion are the respiratory bronchioles, which are those specialized bronchioles that actually conduct some gas exchange, the alveolar ducts, and the alveolar sacs. One condition that is due to structural and functional changes in the gas exchange portion is emphysema. So there's a patient on the left and a normal person on the right, emphysema patient on the left. And what's different? Well, you can see that the person's lung on the left looks quite expanded, and there are very large spaces. In this area over here, you can see some very large open spaces that you don't see over here. So what's going on? Here's another image. Okay, again, you can see these extremely large open spaces right here and here. When you look at this histologically, you can compare the normal alveolar spaces here with these in an emphysema patient. What basically happens is that you lose the alveolar walls in emphysema, and you end up with much less surface area in which to conduct the gas exchange. So this severely impacts the lungs' functional capacity. So let's look at the specific structures involved in this. As I said, there's respiratory bronchioles, alveolar ducts, alveoli, and then there's the vascular component, the capillary bed. So what are respiratory bronchioles? These are bronchioles that have a wall comprised with elements of both the conducting system, in other words, you're going to have at least cuboidal cells, and at least one alveolus. So it's going to have both conducting system and gas exchange system elements within it. So here is a respiratory bronchiole. At the top portion of that open space, you can see the wall of the bronchiole, and that's lined by some cuboidal cells, although we're kind of at a low magnification here. But towards the bottom and at the right, you can see several alveoli opening off of that space. So this is a respiratory bronchiole. So what are alveolar ducts? Alveolar ducts are elongated airways with very little wall. Basically, alveoli. It's like walking down a hallway with cubicles on either side of you. That is an alveolar duct. Alveolar sacs are a cluster of alveoli. So it's like a large open space, like a cul-de-sac, where there's houses all around the circle, so to speak. So that is an alveolar sac. So the exchange of O2 and CO2 occurs between the air and blood. In other words, the alveoli itself and the capillaries. So what cells and other structures form that barrier between the air and the blood? It's called the alveolar septum. So the airspace is on either side in this case, and the capillary is within the alveolar wall. So you're going to have a capillary bed within the alveolar wall containing the oxygenated or the deoxygenated blood, and then you're going to have this alveolar septum here, which is lined by epithelial cells and has connective tissue components within the wall itself, like ground substance, collagen, elastin, anything you would find in a normal connective tissue compartment. Now, you don't just have these flattened cells here, you have some other cell populations too, like these, and we're going to discuss all of those. So the very flattened cells are called type 1 pneumocytes, and the rounded cells that make up part of this wall are called type 2 pneumocytes. The third cell here that's not forming part of the wall, it's just kind of skating along the surface here, is a pulmonary macrophage, also called a dust cell. These live in the lungs, in the alveoli, and they skate along the surface of the alveoli and gobble up particulate matter, pathogens, anything that doesn't really belong there. So here's another view of the wall of the alveoli and the interaction between the capillary and the airspace, and really just a little more elegantly put together than my stick drawing. But it has all the same elements. So remember, the type 1 pneumocytes are flattened, the type 2 pneumocytes are rounded, and those two make up the interface between the air and the wall of the alveoli. And then you have the capillaries within the wall of the alveoli, and the gas exchange has to take place across that barrier. So let's take a closer look at a few of these cell types. Type 1 pneumocytes are actually a small fraction of the alveolar population. However, they occupy almost all of the actual surface area because they're extremely flattened and stretched out. That's attenuated. They are the critical interface for gas exchange, and they have energy-dependent membrane pumps for transport and normal removal of vascular exudates that might be in the alveoli. And that prevents the accumulation of fluids within the alveolar compartments. And you don't want fluids to accumulate there because that would interfere with and compromise respiratory exchange. Type 2 pneumocytes are actually about twice as abundant as type 1. However, they occupy only about 5% of the surface area. They can be identified by very rounded apical profiles in light microscopy and the presence of lamellar bodies if you're looking at an electron micrograph. And we do have EMs of this. I've put them all up on Entrada for you. So there are two very important functions for the type 2 pneumocytes. First of all, they secrete a surface active agent, a surfactant, which reduces the surface tension and prevents the collapse of the alveolar spaces. This is analogous to the Clara cells that you saw in the smaller bronchioles. Their second important function is that they are the stem cell population. So they make more cells that will line the surface. So type 1 cells actually are differentiated from type 2 cells. So alveolar macrophages, also called dust cells, are the scavengers. These cells migrate upwards in the bronchial system, so upwards through the tree, through the airways, and enter the mucus layer, and then they just ride along the mucus layer and get swallowed along with the mucus. Not exactly an appealing thought. So they may also leave the alveoli and migrate into the walls, right, the interstitial spaces in the walls. And sometimes they're responsible for these black deposits in the septa, right? And the last cell type are the endothelial cells that line all the capillaries. These are very similar in structure and function to the type 1 pneumocytes. They're very highly flat. So this chart just reiterates what I said before about the number of cells versus the amount of surface area that they cover. And I don't really want you to memorize this. It's just really a demonstration of the frequency of cells in the lung. Now, this is here to remind me to tell you to look at the electron micrographs of the lung that are up on Entrada. So back to that CT scan that we saw earlier, what happened to the alveoli? Why does it have large open spaces? Well, what happens is whatever is causing the irritation, whether it be environmental or allergy or foreign particles or something like that, you end up getting recruitment of leukocytes and macrophages as a response to that irritation. And the leukocytes recruit eosinophils out into the septal walls, and those cells there start making proteases, elastases, collagenases, and break down the septal walls. So that's how you end up with the loss of those septal walls in emphysema. So you get these greatly expanded alveolar sacs, and they have very little elasticity. Subsequent to that, the walls that do remain end up going through an injury response. So cells come out and as an injury response begin making a lot of fibrous connective tissue. So the walls that are left become quite thick and fibrotic and reduce gas exchange even further. So here are the take-home points, and I think you can read them. I don't need to read them to you. And we'll see you in lab.